I think in order to keep going, particularly outside of academia, you gotta have a really solid why. Founding this company has just shown me the power in loving and caring about people and community. My focus right now is community and surrounding myself with like-minded individuals who are doing the work in different ways, but same goals. I just love science for the sake of doing science, for stewarding knowledge, and doing things like this podcast with amazing people and talking about science with impact.
Welcome to the Science with Impact podcast. Here we have conversations with people from all over about how to broaden the societal impacts of STEM. We have an incredible guest here for you today in Dr. Aneesa Valentine. Aneesa, would you please introduce yourself?
I'm Aneesa. I am a computational genomics scientist by training, and I actually met through a mutual friend who does sort of similar work in this space, trying to fortify the STEM talent pipeline and develop the bioscience workforce. I do this sort of thing because I'm really passionate about science impact, particularly outside of academia. It's why I'm a sort of a bioinformatics solutions architect by day, and why I do a lot of science communication and bioinformatics training and enablement work. I'm really excited to be here and just talk freely about this stuff because I think it's so important.
Why should our listeners, who are very interested in broadening the societal impact of STEM, listen to our conversation?
I think folks who are interested in broadening their societal impact in STEM should listen to our talk because we've dropped a lot of gems, I think, that can help folks disentangle their why. And I think disentangling your why is extremely important in this space, largely because it can be a very isolating space. It is certainly a space that will build your tenacity and resilience and force you to develop very, very thick skin. And so I think in order to keep going in this space, particularly outside of academia, you got to have a really solid why. And I think we touched on that really well from both our perspectives.
Can you tell me more about ImpaktBio, please?
I recently launched ImpaktBio. It's a training and enablement company, as you can sort of see up here. This was a workshop that I did with GenSpace NYC, which is a sort of a community biology lab in Brooklyn. They're a nonprofit. In short, I just help folks work with biological data at scale. So that could be other nonprofits like GenSpace. It could be sort of enterprise biopharma at a more corporate business level, or really anyone who's interested. I typically tend to post free resources online too. I have a Substack that folks can check out for bioinformatics training, steal my code, whatever you want with it. But the focus of all this is just to help people do bioinformatics in a way that is ethical, in a way that's sustainable, in a way that generates impactful science.
So I ask everybody this question, but I want you to dig into your earliest childhood experiences. And can you share any indications that you as a young person would end up in the roles that you occupy today?
So, believe it or not, growing up, I always wanted to be a scientist. I am Caribbean by heritage. I grew up in Dominica, did primary school, which would be our equivalent to elementary school, and high school in Dominica. And during that time, I wanted to be a scientist. Like, I loved biology. There was no scientific representation, though. I mean, Dominica is an island of only like 70,000 people on the entire island. So, growing up, it was sort of okay, you're either going to be a doctor, like a physician, or you're going to be a lawyer. Like those were the careers. Though I wanted to be a scientist at the time, I had no clue how I was going to get there. Perhaps it wasn't until I moved back to Brooklyn, because I was originally born in Brooklyn, started my PhD at the Albert Einstein College of Medicine in the Bronx, and started studying computational genomics, at which point I was like, oh, okay, so first off, the only way to answer biological questions is not just reagent into reagent, pipette this thing and do whatever, but you can use computers to answer questions in biology. And so at that point, I was like, okay, I'll just be a computational biologist. But even then, I had no clue I would go on to pursue a career in data science and AI, commercial data science and AI, or bioinformatics, or start my own company. And I think a lot of that was just born out of seeing particular gaps in my area of work and just desperately wanting to fill them. So to answer the question, I always wanted to be a scientist. Had no clue I would end up where I am today, though.
I feel like for our guests who are listening, can you walk us through how you use computers in bioinformatics and genomics? If you have any examples that you can show us.
So this is a series I started the missing semester, and it's intended to fill one of those gaps that I noticed in my work, sort of bridging the gap between wet lab scientists and computational scientists, and really just enabling folks to analyze their own data and feel empowered doing so. So it's supposed to be a technical newsletter. There's code in here, which I'm going to show you. And it all links out to a GitHub repo that has the entire notebook so folks can download that, run the code, run their own code, do whatever they want. This is a visual of a single cell RNA-seq data set. I think here I used just the quintessential 3000 PBMC data set from 10x Genomics. And what this is doing is helping us visualize our data distribution in the form of violin plots with a scatter plot overlaid so you can see the actual data points. The purpose of this is to sort of set evidence-based cutoffs for filtering your data. Because as a computational biologist, we still need to acknowledge the fact that whatever the folks did in the wet lab experimentally might not have been perfect or foolproof. Whatever the sequencing machine did to get us the data might not have been seamless or foolproof. And so we need to make certain assumptions to inherently clean the data or make it usable before we do any downstream analysis. So that's what we're doing here. We filter out low quality cells so that we have a really robust data set to work with later on. So what's here is the violin on the far left demonstrates the number of genes by counts. So imagine the counts per gene in a particular cell, because this is single cell data. Each cell has a total number of counts per gene in that cell. Each data point is a single cell. The violin in the middle is showing us the total counts of all genes in that cell. So this middle violin is not gene specific. It's just, this is everything. These are all the RNA transcripts that are in a particular cell. And then this one on the right-hand side is important. It's the percentage count of mitochondrial gene expression in the cell, which is an indicator of like dying cells or like overactive cells, muscle cells, if you imagine. And that sort of thing is helpful for filtering out low quality cells as well. So, all in all, it gives us a sense of which cells are actually viable. You want to take the cells that are at the highest density in these violin plots. Or if I were to set benchmarks based on the violin to the far left, I would probably take everything from 500 on the x-axis to maybe 1500 on that axis as an indicator of quality cells. And same for the middle, same for the far right.
As these cells are dying, do they overexpress their mitochondrial DNA or is it just more prevalent?
So the inverse, if they're dying, then mitochondrial genes are mostly turned off.
That makes sense because we're turning down the powerhouse and we're dying.
Exactly. And we don't want those, so we filter them out based on this violin to the far right.
How else do you use computers in your genomic studies?
So this is just for cleaning the data, but this is what most folks do, and that's cluster cell types, because folks want to know what cells they're dealing with, right? So this on its own is not super intuitive. It needs to be annotated, but if you imagine instead of these numbers in the legend, I have blue corresponds to monocytes or orange corresponds to epithelial cells, then I can ascertain whether my cell populations are distinct, which can help with things like verifying whether the sequencing process was clean. There's no like cell population overlap, but it can also help map things like gene expression if you expect a certain cell population to express a particular gene type, have a particular gene signature. This sort of PCA generated UMAP can help us determine that sort of thing in tandem with like a gene expression analysis.
Is that how these dots get plotted, is based on their genomic expression?
Exactly.
Okay, so give me an example, like the red blot. What is that showing us?
For simplicity's sake, yeah, let's say the red cluster are T cells. So you might
Okay.
imagine T cells to express very specific immune markers. Let's say the blue blob are muscle cells. You might expect more energy specific genetic markers in that particular cell type. So if you're looking to stratify cell types, then this, once it's annotated, this can help.
This is incredible. Computers are often used in biology to identify where to dedicate the resources. So, like with our example of the cutoff based on the violin plots, we don't want to waste the assays or the analyses on cells
Hmm.
that are dying. With these, we want to make sure that we're targeting the right cells and not the wrong
Hmm.
cells based on their gene expression. Is
Hmm.
all of this kind of tools so that when you go in to do the assays and the culturing and things like that, you know where you're going in, where you're targeting, almost like the way a surgeon uses scans before they do surgery. Is it that kind of thing?
I would say yes. My area of expertise is single cell analysis and predictive modeling. We use computers to do sort of these quality checks, the violins that I showed, to make sure that we have a usable, workable data set. And that's pretty standard, regardless of what single cell data you're working with, whether it's transcriptomics data, proteomics data, metabolomics, any omics data has to go through this like QC step, which we use computers for. A lot of popular Python libraries like ScanPy, SquidPy, Matplotlib for plotting can help us do that sort of thing. But they also help us answer questions in biology. Like if you imagine folks at the bench collect cells at different development time points. So they've got cells from like E1 through E5. And we do single cell sequencing on each of those samples. So now we have single cell RNA transcriptomic profiles for cells taken from E1 to E5. So computers can help us understand how genetic profiles shift during embryonic development. If I were to do that same blob plot and annotate it at each time point, then I can understand how cell populations shift over time based on their genetic profile. So maybe this, I don't know, maybe CXCL1 is upregulated in this particular cell type in at E2 versus E1. And so that's how we can use computers to help us answer biological questions too, as well as doing things like QC and evidence-based cutoffs and so forth.
I love this work that you're doing because it requires so much mechanistic knowledge about what's happening in the cell to be able to predict and model these things. I don't know. It's just
Mhm.
fascinating. I gotta ask now, like, what made you fall in love with single-celled organisms and wanting to do research and computations on them?
Honestly, I sort of fell into it happenstance in grad school. Transparently, I had a rough lab rotation selection process for all my PhD students out there. You typically get 3 rotations. I had to do 4 because I wasn't able to find a good fit in the first 3. The fourth one happened to be a computational genomics lab, which I'd never touched Python or R or MATLAB or any of the things prior to joining that lab. So I actually struggled quite a bit in the beginning, but once I got it, I was like, this is actually so cool. Like, first off, I don't need to wait 3 months for my experimental results. I just happened to fall into single cell biology, but systems biology and modeling complex biological systems is really big in the computational space. Biomedical imaging, you can answer questions about predicting whether a particular cell is a tumor cell using computers. So happenstance wound up here, but there's still so much that we can do.
You shared this incredible idea when we were having our planning conversation about a skills audit process that you went through when you were deciding to leave your PhD program. And I really feel
Mhm.
like it could be so helpful to listeners at any stage in their career, especially given everything going on with federal disinvestment. Taking the time to audit your own skill set and figure out where you are most aligned in industry could be a really
Mhm.
useful exercise. So, do you think you could walk me through how to do a skills audit? How did you do it on you? Do you want to do one on me? Like, I think that would just be really interesting.
I did mine in grad school. I talked about having a really rough laboratory selection process. That fourth lab that I wound up joining, the computational genomics lab, I wound up declaring in that lab, and that was going to be my thesis research lab. So I started doing that work, and it was fine up to a point. I just wasn't feeling fulfilled for reasons that I'll spare you. But on the skills audit, I decided, okay, I'm gonna leave this program, which we talked about at length. This was in 2022. So in 2022, I decided, okay, I'm not gonna keep doing this, but I can't just not do anything. I need to do something. So I decided to leverage LinkedIn in a way that at the time most folks weren't. And I decided to go look up folks who, random strangers, who had jobs that I wanted to do. Because at the time, you'll recall I was interested in computational genomics. So I'm like, I want to do computer stuff naturally. So I was looking at bioinformaticians, computational biologists. The data scientist was very popular back then, not so much now. I quite literally would take note of all the skills required in those job postings, things like Python programming, R programming, depending, machine learning, all the things. And I mapped that to what I knew how to do then from my experience in that computational genomics lab. And I said, okay, I know how to code in R, but this job wants someone who knows how to code in Python. So how do I get from R to Python? And then once I figured out my outcomes, then it was on me to go do research to actually upskill myself. So learning Python looked like doing free resources like Data Camp, which is still ongoing right now, looking up random repos to see what folks were doing, trying to reproduce their code, that sort of thing. So all in all, it was a lot of looking up professionals who were doing the stuff I wanted to do, figuring out the skills that they had, figuring out what I could do at the time and what I was missing to get to the point that those folks were at. And over a period of 2 months, which was a grueling 2 months for me, because I was also preparing for my qual in parallel, I learned enough from the perspective of hiring managers to secure my first role out of grad school, which was a data scientist for a sales and marketing company doing predictive modeling.
You mentioned data scientist with quotes, and you mentioned that the popularity is down. Can you give me some insight as to why that is?
A data scientist was pretty much a computational scientist across a particular domain. So you'd have data scientists in e-commerce, data scientists in sales and marketing, data scientists in real estate, what have you. And they were just responsible for analyzing data in those respective domains, whatever that looked like. And the popularity is down now, one, because AI can do a lot of that analysis for us. The data engineer persona is up because these are the people who are actually building the architecture and focusing on things like cloud compute, which support a lot of the AI platforms that are on the market today. And so there's less demand for isolated data analysis and more demand for, okay, you can do data analysis, but can you also architect pipelines? Can you also do things in AWS and cloud compute? Can you also do things in Nextflow? So the isolated data scientist is not as popular as it used to be. You kind of need to know the infrastructure stuff too now.
While you were explaining the decline in needs for data analysts and the increase in need for data engineers, I've kind of envisioned this funnel where the lower level tasks go up to the more higher order tasks. And it seems like AI is starting to fill that kind of like lower level part of the funnel. Do you think it's a good thing? Is it a bad thing? I mean, obviously, it won't be that black and white, but what's your perspective on it?
From the perspective you're looking at these broad shifts in a particular domain, I think it's great for mid to senior level folks and just businesses in general because they get to downsize, save money because they're all about the revenue bottom line. And AI frees up more senior skilled folks to do a lot more critical thinking work because it sort of takes care of the boring stuff that no one wants to do, or as you put it, the lower level tasks, like very low cognitive effort. So I think that's good. And I've experienced that in my day to day using tools like Cursor and a slew of models to take care of boring programming tasks for me. As far as whether that's a good thing for the STEM talent pipeline, I don't know that I could say that it is. I mean, I've got friends who just graduated with their PhDs in some sort of bioscience area, molecular biology, what have you, and they are struggling to find jobs, even with this advanced degree and years of accumulated domain knowledge. And I think that is really sad because then the talent pipeline dries up, and that's not something that gets replenished overnight. Once your talent pipeline has sort of taken a blow, that's going to take years, decades to get it back to a point where it's thriving. The PhD alone takes 5 years. I see the benefit, but I don't know that it's beneficial for all groups. That's my stance. AI is still not helpful if you don't know what you're doing. Like it sends me in circles often unless I specify, no, I want you to use this package. I want you to write it in this programming language. I want you to set up this particular unit test. So the higher level thinking jobs, I think, are most definitely safe from both a technical and creative point of view. But the lower level stuff, I think, leaves much to be desired. We'll see. Time will tell, I think. If you could pivot right now and do whatever you wanted to do, what would be maybe your ideal role? And let's not think in terms of title, let's think in terms of things that would be fulfilling to do for you. And then maybe we can just create an arbitrary title for that.
Science has been a safe space for me since I was a young kid. I grew up in kind of an unstable environment. And so I noticed that when I would be reading science books or doing science homework, people more or less left me alone because they'd be like, oh no, she's doing her homework, like leave her be. And it's hard stuff because it's science or math. That dual benefit of it being intimidating enough to keep other people from bothering me while I was trying to do the science and being perceived as difficult. And so just having that safe space to think and envision and imagine, I want as many people to have access to that as possible. And I feel like a big disconnect between science and society is just in the way that we communicate.
Mhm.
I noticed that the higher up I got in my science training, the less accessible what I was doing was to the broader public. I wanted to take responsibility for that, figuring out how can I take this high level work that I'm doing and deconstruct it so that it remains accessible to anyone I talk to. My dream role would be a role where I can freely learn new research that's going on and support other scientists by celebrating and sharing the ways that they are taking these very complex technical aspects of what they do and connecting it to everybody in society.
I don't think I've ever heard that articulated in such a way before. And I think a lot of scientists feel that way. These are goals of a lot of scientists, aside from just doing the actual science. But myself included, for a long time, we just don't know how to voice that sort of thing. Before we actually get into the skills, I just want to thank you for creating this space for scientists, for science adjacent folks, anyone really who would probably consider themselves a scholar, because I tell you, it's so easy to just get caught up in like a respective day to day, just going through the motions, shifting your values and morals to align with like a large enterprise company or a particular group of people. And then you're starving the part of you that actually just really just cares about the science, or just talking to people about science, making science accessible. So just you doing this is an act of rebellion, and that brings me a lot of joy, actually.
That means the world to me. And
Hmm.
these conversations give me fuel in life to keep going. These spaces can be so exclusionary and challenging to navigate. And so getting that, what I think is extremely high praise and gratitude from you, just, yeah, thank you for filling my cup.
Maybe let's take a different angle, and I would love to hear what you feel like you're good at.
I think with most people, our biggest gifts come packaged in our strongest or our greatest weaknesses. I was diagnosed as autistic in my 30s. Some of the struggles of that can be set over here for now. A lot of the strengths that come with that is that I can be very focused on one aspect of something.
Hmm.
I ask questions and I'm curious about things that other people often will just kind of accept as a societal norm because I'm not often privy to societal norms.
Hmm.
Pattern recognition is something that's really strong with me. So, those gifts that I get from that diagnosis have been effort and practice translated into what I think is a really good capacity to take information and condense it in a way that's accessible and engaging and interesting. And I try to encapsulate that as much as possible visually. If I can create a data visualization or an infographic or some sort of figure. And now lately, I've been getting into learning animation and
Hmm.
how to take these into 3 dimensions. How do I make this very abstract, heady, difficult to visualize science and bring it into the real world for people so they can be like, oh, this is what it looks like and what it feels like, what it smells like, what it tastes like, and so that they can use their senses to really understand what they're studying. Just like conversing, pattern recognition, visualization, and communicating.
That's the first step, being honest with yourself about what you're good at. And I love that you almost put the chemistry last. Like you were like, these are the
Yeah.
aspects of me that I'm good at, that I'm proud of, that perhaps make me stand out. Because that's what's important. Like, I hate when people talk about hard skills and like over index on hard skills. Hard skills are important, don't get me wrong. I need to be assured that you can do the job effectively. But past a certain point, especially now with AI, how do you like show up in a workplace? How do you navigate interpersonal relations? Because that is like 90% of any job. How do you collaborate cross-functionally? How do you, if it's a technical role, yes, you did the coding, but how do you present these results to diverse audiences such that they actually find your insights valuable for whatever their particular use case is? How do you assess people's needs and position a solution that effectively meets everyone's needs and across like different functional groups? I love your self-analysis. And for anyone listening, that is step one, like being honest with yourself about the things that you're good at, and then extrapolating those character traits, those skills to roles where you can provide value to an organization through those skills that you've identified, through those gifts that you've identified. And that might look like digging up roles on LinkedIn. It might look like just informational interviews with strangers. Like, what does your day actually look like? Do you like your job? How do people navigate conflict in your role? That sort of thing. So, no, I think this was spectacular.
That makes a lot of sense in terms of like if you can make a skill bank almost,
Hmm.
then you can, once you have your list in front of you, you have a list of what you can do. How do you want to live? How do you
Yeah.
want to show up? How do you want to be? Because different career options will emphasize different aspects of your autonomy,
Yep.
of your presence, of your capacity to communicate, and how
Hmm.
often that's leveraged. I mean, there's just so much associated with thinking about what kind of life do you want to lead?
Yeah.
How busy do you want to be? How many holidays do you want to travel during, right? My husband's a recording artist
Hmm.
and he had to make the decision of like, well, do I want to work weekends when everybody else is often not working? And what does that mean for my family life? And how will I
Hmm.
balance that? You know, because he's also an exceptional father.
Hmm.
So thinking about how to navigate these different aspects of who you are and who you want to be, it's important because you can find yourself in the role that leverages all your skills and talents,
Hmm.
but you're miserable. It doesn't honor who you want to live as, you know?
1,001%. There's a lot of identity work that goes into starting a PhD program or just navigating doctoral training. The training is one part, like the hands-on skills that you build, but far more important, I think, are questions like, why am I doing this? Am I doing this because I anticipate the degree helping with my career progression? Am I doing it because I want to prove something to myself or I want to prove something to my family? Am I doing it simply because I love science? Because those questions really impact how you perceive success I think it's just so unfortunate that folks don't start asking themselves this question until they've graduated and then they're struggling to find jobs. And then they're like, well, why did I even do this in the first place? Is this really what I wanted to do? Or when, for instance, you might have wanted to do research in an industrial setting, but you are only getting interviews to be a project manager, for instance, or a product manager or something. And all of a sudden you're like, well, how do I did this degree for 5 to 8 years. How do I now transition into being a product or project manager? And then sometimes you even find out that this is a better fit for me, not because of the hard skills that I've developed during the PhD, but because of who I am as a person. Yeah, I know how to pipette, but I actually just love communicating with people. And it turns out that being a project manager was actually a much better fit for me. So, all that to say, do the work, do the inner work. If you're a prospective student, do the inner work now. If you're a current student, actually do it now too. Do it before you graduate effectively. It'll save you a lot of grief, I think.
If you're finding yourself getting angered or saddened or unfulfilled, there might be something there to tug on and
Yeah.
explore. Let's say as you move up the career ladder, if you're finding yourself communicating with your subordinates in a way that's harsh or unprofessional, even, there's still a lot of inner work needs to be done there. And I think that the era of favoring technical skills over people skills is
hmm
quickly dying off. As someone who founded your own company in ImpaktBio, what has that entrepreneurial journey been like for you? And what have you learned about building an organization that's focused on science for the people?
Impact Bio was intended to be a training company, and it still is, but I was not thinking about scale in the very beginning. It was intended to be a sole proprietorship that would allow me to do other things, help nonprofits to train their workforce, help enterprise biopharma to train their R&D scientists, but it was very me centered back in July when I registered the company. Founding Impact Bio has allowed me to meet so many different people doing so many different things who are also focused on science impact, people like you. I've met so many amazing people, and founding this company has shown me how important it is to just love people. I love community. I love talking to people who also love community, and thankfully, founding Impact Bio has allowed me to be surrounded by a lot of those people who just care about community, doing different things, but founding this company has just shown me the power in loving and caring about people and community. And at this point in my career, which honestly hasn't been that long, but still my focus right now is community and surrounding myself with like-minded individuals who are doing the work in different ways, but same goals. So it's turned into a real people and community company, actually.
I love that for you so much, Anissa, because I'll tell you, I founded my company the same way. It was out of necessity and it was an urgent need when I founded what was the original name of this company that is now Science with Impact. As my dire need to feed my kids was covered and I felt secure there, I felt myself open up to how much this company could serve others and how much good I could do. So, in our closing tradition for this podcast, we have our past guests ask our future guests questions. Jeff Kozlovsky is asking you this wonderful question.
I would ask Anissa if she were to start a podcast, what would it be called and what kinds of people would she interview?
So a little bit of a backstory and context here. I recently came across this newsletter. It's called Forgive Me to the Folks if they ever actually hear this. I think it's called the Scholarly Newsletter. Two folks who really care about science and just talking about science. And they decided to create this newsletter that talks about just doing science for science's sake. And they've got this piece called the Scholarly Manifesto. I have it saved in my phone because it was just so profound for me and helped me understand a bit more of my purpose and my calling actually in this space. But the long and short of it is that they make two distinctions, one between the academic and the other being a scholar. The academic is someone who, by no sort of reason of her own doing, she has turned science into a product, like something to be manufactured something that is attached to like certain success KPIs. So they talk about the academic who basically no longer does science just for doing science's sake. She does science to be published in these high impact journals, to be cited, to be recognized. Whereas the scholar is someone who does science just for doing science's sake. Like the scholar is in pursuit of knowledge that is going to better humanity. She doesn't care about being cited. She doesn't care about making money off of a scientific product or anything. She's just doing science because she loves, this is the language that they used, stewarding knowledge, which I think is part of my purpose and calling, actually. And so, all that to say, if I were to start a podcast, I would call it the Scholarly Podcast because I do consider myself to be a scholar. I just love science for the sake of doing science for stewarding knowledge, and we assume almost a duty of care for the knowledge that I steward through ImpaktBio, through my Substack that we looked at earlier, and doing written content, through doing things like this podcast with amazing people and talking about science with impact. So I would call it the Scholarly Podcast. It would be by scholars, for scholars. That's my spiel.
Well, speaking of people who bring joy, I don't know if you know Vernetta, but if you don't, I'm honored to introduce the two of you by way of your question. I'll give you a quick overview of Vernetta. So she's also an academic entrepreneur. Her background is in writing and in composition, and she got her doctorate in that. And now what she does is that she helps researchers of all kinds to write their dissertations through her company. She is so phenomenal, and I'm really honored to get to introduce you. When you're ready, Aneesa, let me know what question you'd like to ask Bernetta.
I'm curious to know how she curates her learning material in a way that is receptive for diverse audiences, because it seems like she's done work across a ton of different domains. With a doctoral degree in English, I would be very curious to know how she presents her material in a way that's receptive to scientists, to engineers, to administrative folks, like that sort of thing.